biotechnology has primarily two main aspects. One is called genetic engineering. The other aspect of biotechnology is maintaining aseptic conditions. Genetic engineering. Generally, when we hear the word engineering, what immediately comes to our mind? Engineering is a course. No. You look into the dictionary. Engineer means bring about a change. So genetic engineering is the technique of bringing about a change in the genetic material of an organism. To bring about this change in the genetic material of the organism, you need a situation where it has to be done under very specific aseptic conditions. Genetic engineering, therefore, is defined as in vitro manipulation of the genetic material DNA. We are already familiar. The word in vitro means outside the body. That means DNA is removed from the cell and is altered, manipulated. The term manipulation indicates that we are going to bring about a change to suit our needs. So, in vitro manipulation of the DNA. And once this is achieved, this DNA, manipulated or changed DNA, has to be introduced into a particular cell, a host cell, and that host cell is to be cultured under totally aseptic conditions. This in vitro manipulation of DNA has 3 steps. First one is identification and isolation of the DNA. DNA of which organism is supposed to be manipulated. Identification and then isolation, removing the DNA from the place of its occurrence. Identification and isolation of the DNA. Introduction of the desired gene. We are not interested in transferring the entire DNA into another cell. We are interested in a particular gene. So the gene that is supposed to be transferred into a new host cell, that is called desired gene. So introduction of the desired gene into a new host cell. And number three, maintenance of the transferred gene, and its expression in the progeny of the host cell. So you are going to identify the DNA that is supposed to be transferred. From that you are going to isolate the desired gene, transfer this desired gene into a new host cell in which this DNA is not present earlier, maintain the transferred DNA and see that when the host cell reproduces, the transferred DNA also reproduces or replicates. So these are the three steps that are involved in genetic engineering. The maintenance of the transferred gene and its expression in the progeny of the host cell requires maintenance of the host cell under total aseptic conditions. So this is achieved using a technology, and the technology used for genetic engineering you can also call it as gene manipulation, This technology is known as recombinant DNA technology. Recombinant DNA technology. It is because you are going to recombine DNA obtained from two sources. Obtained from two sources. One is the desired gene, which you are supposed to introduce into a new host cell, and the other one is the vector DNA. You require a carrier. You require a carrier DNA for transferring this desired gene into the new host, that is called vector DNA. So you are going to combine desired gene and the vector DNA, and the resulting DNA is called recombinant DNA. Therefore, this technology is known as recombinant DNA technology.